Yes, ma'am.
A very good morning to all. Today, we gathered here to celebrate the World Animal Day. This day is celebrated in 1925. It's almost a century now. So I welcome you all, my fellow colleagues, students, research fellows, our neighboring school students, teacher, our distinguished guest, and our honorable director, ma'am. So before we get into today's business of listening to the distinguished lecture, I will invite our director, ma'am, to say a few words on this very day.
Thank you very much, Dr. Bapaditya. Very good morning to one and all. Esteemed speaker of this day, Professor T.P. Singh, my dear colleagues, students, fellows, and school children, and the escorting mentor of the students who is amongst us on this very important day, which is the World Animal Day. As Bapaditya rightly said, that it was 1925 when this day was starting. This day started. So yeah, next year we will be celebrating the completing 100 years of it. It's the 99th World Animal Day, and for this day, me not only I am very happy and very excited to hear Professor Singh. All of us, we were talking, discussing, students and my colleagues. Let me just tell you, when we met last month at Vadodara, and it's always so good to hear Professor Singh. It was early 2000 when I was like you, and we invited him at IVRI, and that was the first time I heard, and then there were many occasions. And the moment I requested, he was so generous to accept it immediately. Thank you very much, sir, to be amongst us. And I, on my own behalf and on behalf of NIB fraternity, welcome you. So we celebrate this important day through the wisdom and the data which you're going to share with us. He's a known structural biologist. I think I'll leave it on Dr. Bapaditya Day. With that, we look forward. Have a wonderful day. Greetings of this important day. Thank you.
Thanks, our director, ma'am, for the brief introduction. So now I formally introduce our distinguished guest, Professor Tejpal Singh. So it is a both honor and privilege to introduce to you a luminary in the field of structural biology, a distinguished Science and Engineering Research Board of India Fellow at the All India Institute of Medical Sciences, New Delhi, and a true global science leader, Professor T.P. Singh. Professor Singh's journey through the intricate world of science is nothing short of remarkable. With a master's degree from the University of Allahabad and a PhD from Indian Institute of Science and a postdoctoral fellowship as an Alexander von Humboldt Max Planck Fellow in the laboratory of Professor Robert Hoover, a Nobel laureate, that he embarked on a path that would lead to transformative breakthrough in structural biology. Returning to India, Professor Singh served as a reader at Sardar Patel University and later joined the Department of Biophysics at the All India Institute of Medical Sciences in New Delhi. Here, he initiated pioneering research program that would redefine our understanding of protein structures, peptide design, and drug development. With an illustrious career spanning several decades, Professor Singh has left an indelible mark on the scientific landscape. His contributions include determining the three-dimensional structure of numerous proteins, and I counted more than 600 of them. And some of these are lactoperoxidase, peptidoglycan recognition protein, lactoferrin, ribosome inactivating proteins, and many more. These accomplishments have not only expanded the boundaries of knowledge, but have opened doors to innovative application in medicine and biotechnology. Professor T.P. Singh's commitment to advancing the field of clinical proteomics at All India Institute of Medical Sciences is poised to revolutionize our ability to understand, diagnose, and treat a myriad of diseases. His relentless pursuit of scientific excellence has earned him the distinguished fellowship of all three major science academies in India and several globally. With more than 11,000 citations to his name, Professor Singh stands as a true giant in the realm of structure biology. His contribution to science have been recognized with a plethora of national and international awards, including the Distinguished Biotechnologist Award by DBT, Professor G.N. Ramachandran CSIR Gold Medal for Excellence in Biological Science and Technology, Sir J.C. Bose Memorial Award, and many more. And he's also the proud father of a popular shark of the famous Shark Tank program. Many of you might have seen it, watch it on television. Vinita Singh, who once quoted on a popular social media forum that the irrational passion she has, it must be genetic. And that the same passion Professor Singh has infused into all the researchers he trained and inspired over several decades. Those of my colleagues who are from Delhi or did their studies, masters or PhD from Delhi, we had the privilege of attending many of his classes, structure biology classes in AIMS. So as we gather here today, we have the distinct privilege of learning from Professor Singh's wealth of knowledge and experiences. His scientific lecture promises to be a journey to the frontiers of structure biology, offering us insight into the observed differences in the expression, structure, and function of the innate immunity protein in different animals, which are indicative of their therapeutic potential. So without further ado, let us warmly welcome Professor Singh. Sir.
These were not a good choice, Taru, to call him. He said so many things, and these so many things are not to be said. Bhavati, why don't you sit down? No, I think we should not talk about people. But thank you so much, and thank you so much, Dr. Taru Sharma, that we met in Vadodara just like that in the conference, and she mentioned about this animal day. I never heard about this, but this is a great thing that there is something like Animal Day. I think to me and to most of you, animals are like gods. I consider them a big opportunity in life because of them. And for the last many years, to be precise, more than 3 decades, I have been working on animal proteins. I'm very much part of your DBT institution, but I have been interacting with the ICR Institute for so many years. I have been to all institutions, collect samples, collect all kinds of samples. So my whole career, not whole career, a major part of later career is on animal proteins, and they are fantastic. We never realized this before. There are many things we did not know before, and you will see that how important they are and why we have been missing them. But before I go to talk about what I will say, I'm really thankful to you, Taru, to invite me to this place. It's a remarkable place. I heard about this place quite many times. And Dr. Bhan, who you have put this auditorium on his name, I'm so thankful. He was my colleague. And when he was selected for DBT, he came 3 times to my office saying that, should I join or should I not join? He was wondering that what a clinical man will do in DBT. And if you become a clinical man in your life, the patients and OPD, you consider yourself incomplete. So he was in dilemma. He was arguing so many times. I said, do it, because you can do many things which many people might not have thought about. So he joined, and I think it was a great opportunity for DBT and for all of us that he became secretary. Many programs he started. He was so enthusiastic. We all are benefited by his things. So DBT has made great progress during his time, and of course, DBT has been doing very well subsequently, but his contribution enormous. So I thank you very much. So there's so many reasons for me to be very excited, to be happy that you invited me to. First thing, animals, very important for me. Dr. Bhan was my very dear colleague, and you are my very dear colleague. And I also find many, Dr. Havde is a wonderful friend of mine, and many students who have been, he never disclosed. He didn't disclose it. And many students who were in South Campus or JNU, they are here. So I feel very, very attached to this place. Thank you so much. And I'm very happy to be here. So I will share some of our work. I think we can have less slides, then slides will look better. The structural slides look better when, oh, this is forward. I think I'm going in the reverse direction. Yeah, I'm experimenting. Which is backward? This is backward. So I go, okay, I come to this. And this is the pointer, is the top one. Okay, that's why the mistake was. I was single pressing when it's pressed long time. Then it becomes a pointer. That's a trick. Thank you so much for the trick. You see from the title, you can have less light still. You will see them better. Is it possible to come there? I think I can have this because if this works, that's the best. Oh, very nice. Okay. Good. Hello, can you hear this? No, he has to put this device.
Keep this in proper.
Huh?
Please keep this in pocket, sir.
Pocket, any pocket.
Right.
Okay.
Switch off everything.
Everything switched off? No? Okay. That is working now. I can go there. Yeah, I have to see that. I should not fall before I finish. Okay, so I want to be closer to all of you. Well, I cannot see from that angle. Is very. No, you can switch off some of these lights. This also. Okay. Only these ones less.
Yeah, this is good.
I'll go to that side. This is a better way to look from the right-handed system. You see, you can read from the title is so have less. This is better. Just stop it here. Okay. All of you can read these slides have very small text and they are boldly written. You can read from the text that I'm so grateful to this animal kingdom. And I think all the people here working here will be equally very happy to see that. And I think many of you are working on bacteria and viruses which affect animals. And animals also include humankind. We are all animals. So everything is applicable to all of us, including your cows, buffaloes, cows and goats, everything. So I'm still trying to, okay. So you all know when we talk about these infections, I'm also addressing these young boys and girls, school girls. So I will talk at that level. When you talk about infections, these are caused by many organisms, and primarily, more commonly, we know bacteria and viruses. And we all know about coronavirus, all of you. It was a devastating. People complained about coronavirus. Lots of people lost their life, unfortunately. But I will say it again, and I think many microbiologists would agree with me. Viruses and bacteria are not our enemies. I think they want to use us for life. And if we can support their life, they are happy, we are happy. But unfortunately, we succumb because our bodies are not well prepared, not well kept for looking after them and ourselves. So, but anyway, you know what happens. Here is the common entry point, particularly in reference to the coronavirus, which all we remember so well. They all enter through your nose, mouth, and eyes, and any other opening in your body. Now, wherever they enter in these sites, at all these sites, our bodies have certain proteins expressed. And these proteins are expressed in response to external pathogens. The sites they appear, they enter, certain proteins are expressed. And these proteins recognize them, that's how they are expressed. Their expressions go up. Now, why do they are expressed there? Because they interact with them, they neutralize them. Theoretically, every organism entering our body should be neutralized by the proteins expressed at those sites. And this must be happening all the time. You see, sometimes we succumb to this situation that we fall sick, or sometimes we die also. But that's a very rare situation. Most of the times, these proteins which are expressed at those sites are fighting and succeeding. We don't come to know. We only come to know when there is a crisis. So our bodies are all ready to fight them. But if something is much more massive, then we are not able to fight. So, but once they come to our body, they create other antibodies, other immune-related proteins to make us more protective. In other words, all these organisms are recognized by certain proteins. And the first recognition is done by proteins known as the proteins of the innate immune system. All of us have a germline innate immunity. The antibodies and vaccines, they appear later. But the first line of defense is provided by innate immune proteins. They're all there. And theoretically, they should protect us all the time. And they also protect us all the time, except that sometimes we come to know that they are not able to protect. So we don't recognize their strength, their support, because we don't notice it. It's happening all the time. So this slide just to give you an idea that certain this kind of infectious diseases which enter our body through air have been very, very difficult in the case of coronavirus. The whole world was affected by this. Lots of times people say that certain countries are much more advanced than other countries, and they should have been able to protect themselves, but they were affected the most. There were no known medicines against this virus. There were no vaccines. And then at that time, you would talk about vaccines. Some people say that vaccine development takes 10 years. But I think this time this was done very rapidly. Of course, we don't know many things about what vaccines did, but we were not prepared. That was the point. But what we didn't worry about is what we will see today. Why didn't we re-examine our innate immune system? The first line of defense, why it was not able to protect us. Where the other weapons will happen later. Vaccines will take time, antibodies will emerge after a while. So that is a question, and that question we should examine here. Now, when you fight infections, fight these invading microbes, how do we fight them? The first and foremost is your innate immune system, this one here. This should be able to protect us. It's like a border security force that all the time some kind of skirmishes are going on, but the border security people protect us and nobody enters. So they should in principle protect us. And of course, after a while, after some time, specific antibodies are made against those pathogens. And in the meantime, even vaccines emerge. This time they emerge very fast. Normally they take longer time. And of course, we are very fortunate. Many decades ago, some very potent compounds were discovered, known as antibiotics. And the last one, which were made in the lab, some synthetic antibacterials were made. So these are the 4 ways we fight infections. And we have been successfully fighting in most cases, but some cases become pandemic because this is a massive thing. And all our things are not able to fight, but we should ask why. Now, this slide is particularly, I think we should have some perspective to historical situation. Many centuries ago, when we had no idea, now we talk of a lot of things, vaccines, antibiotics, even innate immunity we talk, and synthetic antibacterials we talk. What was happening when 2,000, 5,000 years ago? There were people living in this world. There were these organisms, and they were causing infections. How were they fighting? And you see, if your mind is trained means it's logically able to think logically, then this part of the world where people were more civilized, they were thinking, their mind was functioning in a positive way, they could think about certain materials. You see, if you read all these, they identify certain materials by way of doing experiments. They tested these materials on cases, and they found they were curing patients at that time. They had no idea what these materials contained. You can see this, all these kinds of things. You can read it for yourself. So these were just by way of observation, they found that these materials were protecting them against infections. Now, if you go to the next slide, you see that this geography, which is here, this is where civilization occurred first in this part. The point is slightly more flexible. So what I mean is this region. You can read this location. Okay. Now you see this geography has changed. But if you see the first one, if you can read this, recommended using mold for treatment. The mold which were identified in previous locations. They had no idea what it contained, but these people, where technology emerged first, industrialization occurred, they started from using the materials prescribed in the previous locations, which were identified by way of observations. And then slowly how these things grew. You can read those things. And somewhere you read also that they found that bacteria were killing bacteria. It means bacteria contained something which was killing another bacteria. And then you find that some fungus. Can you read this? Penicillium, it was a fungus. So there was an experiment, a guy was culturing, and he had a cultured plate. He left the plate open and went on a vacation. It's a very strong habit of Europeans and Western people. The vacation is as important as anything else in life. So he was in a hurry and he left his petri dish open. And when he came back, part of this was covered by fungus. But what he noticed was wherever there was a fungus, culture did not grow, bacteria did not grow. So it struck to him. That's why it's important to be a trained mind. Somebody will not notice this and throw the petri dish, but he thought that this fungus contains something which is killing bacteria. And that was an important thing. Therefore, education and training in science And technology is important because trained minds cannot observe things, can absorb things. So he immediately realized that penicillium, what it calls this fungus, contained something. This was in what? 1928, somewhere here. That this fungus contained something which was killing bacteria, but he didn't know. He was a microbiologist, biochemist in those years, or microbiologist. He didn't understand. He observed that fungus was killing bacteria, but he had no idea what it contained. Then he went to a chemist colleague next door and requested him, can you analyze this fungus? And then he identified a compound, penicillin. That was the first antibiotics. And that's how antibiotics emerged. Once you discovered one antibiotic, then people started isolating more such compounds from these organisms and then started modifying them in the lab to make more antibiotics. That's how antibiotics were discovered. And for many decades until now, we are well protected against many diseases. So antibiotics have been a revolutionary, a great achievement of mankind to fight against these organisms. So that was the status at that time. And that is why many scientists in the labs and pharmaceutical companies did not develop synthetic antibacterials. That was a really pity. And you see that some sulfonamides were made as early as 32 and then gramacity, very little. Because antibiotics were so effective, so potent, there was no need to do anything else. So that is how the science grows. You know, when you are in crisis, you do faster. When you are, she was using the term that comfort zone, we get into a easy life when things are looking good. But then after many, many decades, we observe that I think sometimes in this part of this period, they came in 1928, and by the time it was 1990s, all of you are following this, or you're sleeping? This is the most important population today in this hall. Is it making a sense? If you understand, then everybody will understand. If you don't understand, then nobody will understand. So that's how, so then you might have heard about the resistance by bacteria to antibiotics. Although it might be happening before, when you use certain compounds continuously to kill these organisms read about these compounds, and then they modifies the sites where these compounds bind, and they change those sites by, these are mainly proteins. They mutate those proteins at the binding site, and by way of doing mutations, they don't allow these compounds to bind. And that's why you call this mutation. So the bacteria did this. Bacteria and viruses, they all do all the time to survive. Everybody, every living organism has to survive. So this is how it goes on. So now we are back to a situation as if we don't have antibiotics. So we have to think about new substances which can protect us from these infections. Antibiotics are no longer potent enough, or we have to modify them in such a way that they are able to bind even to the mutated sites in these proteins, in these organisms. This might be too preliminary to many people here, but I think this audience in the middle is the focus. Is that okay? Yes, somebody said yes, very neatly. So now, coming back to this again, now you have a problem that antibodies take a while after infection. There are specific proteins which neutralize the proteins of invading microbes. That takes a while. By the time, if there is nothing, you succumb. And then vaccines take time. This year, but this time in this pandemic, special rules were used and relaxed, and that's how vaccines came faster. But every time this doesn't happen, many scientists complain. And I remember that when that human trials were skipped, some letters came to us also that you don't worry, in ethics committee, just clear this. I was of this opinion that we should clear this, but many of my committee member said, no, you can't do this. But I think we should realize that we should do this. So we did it finally. So this takes time. So you have this time you cannot reduce until you have made some. You can't have an idea as to what this virus is, unknown virus, or bacteria if this is unknown. And antibiotics are not working. So these antibacterial was not there. Why didn't we think about innate immunity? Why innate immunity is not able to protect us against this difficult virus? I wouldn't say difficult. This virus, no virus is difficult. It's just unknown to us. Why innate immunity could not fight? It is fighting all the time, protecting us. But why in this case it could not fight? It must have been a very massive loads of the virus very quickly, and your innate immune proteins may not have that level. It's a general way of protection, may not have that kind of potency of capacity to neutralize them quickly, and it spreads over, and that's how it was happening. So we should ask a question: is there a way that we can think of improving the quality of our innate immune proteins? Most of the people think that innate immune proteins or innate immune system, there are no role to play. When I first made one seminar in IISc Bangalore many years before, and I said that innate immune proteins could be used as therapy agents. One guy from UK said that I didn't hear about this concept. Is this feasible thinking? So every thinking initially may look not so feasible, but you will see that how feasible it is. So we should see what we can do with this. And your animals, I think NIAB is a fantastic name, animal biotechnology. And your animals can provide solutions to many things. Now, when you talk about this particular innate immune environment, so you see that the protective functions of secreted components from epithelial cells, the cells which are at these sites, at sites which face these pathogens. Now you can see there's so many ways we try to protect us. This all is a innate immune system. This all is the first line of defense. You have so many ways that there are some physical barriers, and you can read this. What is this? Direct, and this one, this next one. Produces certain compounds which are that, and then you can see, and then you can see. So these yellow colored things are the ones we have studied, and we found some ways how this innate immunity can be exploited. Now we worked at large number of proteins, but we started these animal proteins to begin with. Unlike today that we think there's a problem and we should solve it, when we started protein structure determination, the main idea was that to do a structure of a protein. Doing a structure of a protein in 80s and early 90s was a real major goal. So we started proteins, and I must tell the story to Taru, you must know this. When I was trying to look at doing structures of proteins in those years, there was no expression system. Today you can express proteins in high quantities, clone, express, and produce a lot of quantity. In those years, we used to be dependent on natural proteins. So I was trying to study some plant proteins, isolating them, very low concentration, keep on isolating, keep on running long columns. Then I found a colleague from NDRI Karnal. We had 2 electron microscopes in my department in those years. There was no structural biology, but electron micrographs were there. One colleague from NDRI Karnal, his name is Dr. K.L. Bhatia. He was passing in front of my office, going back and forth. I wondered who is this old gentleman. I asked him, what are you doing? He said, I've come to do some electron microscopy of my protein, which I have isolated from buffalo. I say electron microscopy at that time, not the cryo electron microscopy of today, cannot give you anything about protein molecules. I said we should purify this protein, crystallize into the structure. And that's how my journey with animal proteins began. This was with NDRI Karnal to begin with. And of course, I have gone many times to your place, IVRI, and I have gone to all the ICR animal institutes subsequently. So this is a story. So I will talk about little bit about this thing, two simple proteins of this innate immune environment here. See, these are the four members I picked up whose structures we have studied in great details. So they all some way responsible to protect you. You see the first one, the peptidoglycan recognition protein. It's a protein which recognizes bacteria and other organisms by recognizing their cell wall molecules, which are different from humans, don't have cell wall. So that's the mechanism. Once it recognizes path, binds to them, and then subsequent things happen. And of course, this protein sort of it binds to iron with very, very high affinity. 10 to the power minus 20 molar affinity it has from the environment. And that's how it deprives bacteria. This binds to cell wall molecules and then neutralizes bacteria and viruses. And this lysozyme, of course, everybody knows it hydrolyzes PGN. And this particular last protein, lactoperoxidase, his name is lactoperoxidase. People may think it is present only in milk. This is present at all the sites, in all the mucus. Wherever there is the bacteria can enter a body, this protein is present. And this protein works as an enzyme. With the help of hydrogen peroxide, it converts thiocyanate into hypothiocyanate. And this is antiviral, antibacterial. This kills bacteria. It's a very simple thing. So these four proteins, if you see, why cannot they protect us? And we should see how can they protect. Now, this is your wonderful animal kingdom, I think. Perhaps many scientists here are the modern scientists. They don't see animals, they only clone express proteins and use animals as experimental animals. But animals are very important. See, I divided this into two. One, they are the domesticated ones in simple environment, domesticated ones and live in normal environment. But then we thought these three are living, this is very hostile, warm conditions. This is not a very clean environment. And this fellow lives in a very cold environment. The question here is that the proteins of the innate immunity, which are expressed in animals, including humans, who live in normal environment, temperature, weather conditions, pathogen load, all those things are protected somewhat. And those animals which live in very hostile conditions, where there are very strong bacteria and viruses, survive high temperatures and low temperatures, and they in fact, do they affect these proteins differently than the ones which live in normal environment? This one. So this was one question came to mind, and that was a very interesting question. See, now the first thing is that look at the concentration. Two ways to judge how these animals are expressing these proteins. There are two things. These proteins, which are responsible to protect us, how are their concentrations? If their concentrations are good, they can fight more. If you have more weapons, you can apply them more. And how are their potencies? These two arguments. The first and simple thing is, you see the concentrations. These proteins, human proteins, all these have normal concentration. This particular concentration protein has a very low concentration. Similarly, these also have a similar situation. And you see these two animals have special this, and but rest of them are very low. So this variation, does it mean anything? The first and foremost thing you should see how the concentration of a given protein is in different animals. That will speak about that this animal is dependent more on this protein, and where other animals are not dependent so much on this protein. So we should check what is happening to this protein, and we will check, of course, this protein to see what we can do. So does this make a point sense that if a protein is in a good concentration, if you are eating bad diet and you don't have amino acids and your body is not able to make proteins, you will die when infections occur. But there should be a demand, there should be a machinery to synthesize proteins. There should be a trigger, there should be an initiative. So in this case, in these two animals, this particular protein is in high concentration, this one here. So it's not investing on these proteins. These two animals are investing in this protein. Other animals are using all these proteins, but not investing so much on these proteins. So it is something like that, that a country is making weapons, guns and tanks and so on, and one country is making only what? Nuclear bombs and nothing else. So that is how you can see this. It's the same way our body is really protecting. These are our enemies, to say so. So we should protect like this. Now I've just to give you very simple and very quick. I'm not going to spend so much time. This particular protein, which is expressed in high concentrations in camel and porcine, I thought this will be expressed likewise in yak. It didn't happen there. So cold conditions are not that hostile, but hot conditions and dirty environment are critical. So we'll just quickly see this protein. What is the difference in this case? In this particular family of protein, there are 4 members. These 4 members, but these are common member, which is recognizing the bacteria and viruses by interacting with the cell wall molecules of these organisms. So we will just talk about this because that's different, all 3 of them. And the next steps are other steps of hydrolyzing and killing them in some mechanism. Now, the first and foremost thing, as I said, that this particular protein, we saw the concentration. These are basic things which should illuminate you that if something real is happening. You see, this particular protein, if you see humans, is this peptidoglycan recognition protein, very low concentration here in humans. If you come to this porcine, the very high concentration here. This can be isolated from tissues and mucous membrane, but those sources give you little because this is very little amount. So we did this. It's also expressed by mammary gland. So you can see from colostrum and milk. So this protein is high concentration colostrum of porcine. And if you see in camel, also this is normal. Milk also has a good concentration here. But you see bovine, buffalo, yak, goat, sheep is very much missing here. So first thing that this particular protein, which fights these organisms by recognizing cell wall molecules, is expressed highly in porcine and camel. It is not expressed in other animals. So that is the first step. Now, your mastitis, madam, is an issue. Every time Dr. Yadav called me and said that we give diclofenac to our animals and then vultures are dying because the animals are dying and vultures are eating animal flesh and that has a lot of diclofenac and that's a poison to vultures and they're dying. They raise this issue. Can we do something to protect animals who suffer from mastitis, massive disease? And I also read in literature, I think you all know that, there's so much loss of milk because of this infection. So you see that when this is infection, this particular protein is low, normal times, low concentration. But even in these animals, this concentration goes up. This is for camel, of course. The concentration goes up. So mastitis infection induces the concentration of this protein so that it can fight better. Now, so you have now example of, let's say, human, camel, and porcine. Human protein normal, very low concentration, but human protein. And I compare with camel, where concentration is high, of course, and concentration is high. The first step is to compare the sequences. Do they indicate something? Do the sequences of these this protein of these animals differ in some way. And we compare, very few differences over there. These are very critical ones, but let me not go into this detail. And the consequence of this, you will see in the structures. And okay, this can be skipped. When we see the structure, in human, this protein exists as a monomer. You see, protein molecules in your body and in any living organism can function as single molecules, monomer, what we call, or two molecules together produce the biological effect, sometimes three molecules together, sometimes four molecules together. By combination like this, they enhance their capability of biological functions. You understand this? So this is an important indication that why a protein in humans is monomer, in porcine is trimer, and in camel is tetramer. The fact that the organization of these proteins are different, their effect of biological function, the potency of biological function will also differ. And that is primarily residing in their amino acid sequence. And there, in this case, there are only few mutations, few changes. It's not so much change. Very few critical amino acids, at the most two, have changed this state of oligomerization. So this is what we call state of oligomerization. The protein functions as a monomer. The protein can function as a dimer, as a trimer, as a tetramer, or a multimer. And these things are generally done by systems, these organisms, to improve their biological function, to alter their biological. So this is a structural indication that the proteins from these three animals show different structure organization. The potencies will differ, the effect will differ. So if you see now, let us look at this camel protein and compare with the human protein. What happens? This is a human protein, in a very simple way. And in proteins, proteins are globular molecules, and all the proteins have a cleft where the other compounds, other molecules bind. So here also you see there is a cleft-like thing here, and the protein is folded in this way. This is a human protein. Now, camel protein, you saw 4 molecules. Now, if you superimpose the single molecule of camel on this human protein, or let's first see how the bacterial cell wall molecules bind to this. You see, this is a protein, human protein, and lipopolysaccharide is a cell wall, one of the cell wall molecules in some bacteria. You must be studying biology, some bacterial cell wall, lipopolysaccharide. You heard this? So lipopolysaccharide is recognized by this, and you see how it is held with this protein. Even two molecules interact, when two large molecules or two molecules interact. Now, how will the binding affinity, the potency of these proteins will determine as to how many interactions it makes with the binding molecule. And more the interactions, higher the affinity, higher the activity, isn't it? So if these two compounds, this protein and this compound, if they have large contact region, and as a result, will make more interactions, it will be higher affinity, it will recognize the cell wall molecules much quickly and will neutralize rapidly. If it is doing that faster, the bacteria virus cannot multiply. So that affinity is very important. So this is determined. Now, these some interactions will occur here, the rest of the molecules are lying here. So similarly, see, this is lipoteichoic acid, also cell wall molecule, and PG and peptidoglycans also cell wall molecule. This is how the human protein is interacting with these cell wall, selected cell wall molecules of bacteria, for example. Is it okay? It's not like your class, something similar to your class. It's similar to the class in the 11th and 12th. The rest of the people must be very worried about this preliminary level of discussion. Nevertheless, now let's see the camel protein. It's superimposing. Structures are identical. The only thing has changed is that camel protein is now in the form of tetramer. Otherwise, individual structures are identical. It didn't change the structure, It only changed the oligomeric state. And then you see, since it is a tetramer, how does it do that? Now you see when you have second molecule here. Now these compounds are much more buried here. There are more interactions with the dimer than the monomer, isn't it? More interactions means better recognition, faster action, more successful in protecting. Is this clear? If you can say yes, it's like poets, they demand clapping. If you can say yes, then it makes sense. Okay, now if it's a trimer, which is in the case of porcine, you have still more interaction here, buried here. But in camel's tetramer, then you have this completely buried now, these films. So as a result of oligomeric state, camel and porcine proteins have become far more potent than the human protein. Now the question is, why can't we use camel and porcine protein for humans to protect against these infections? There are two ways that either you can use them directly, because antigenicity is not there, it's the same protein, except some mutations which are buried in the dimeric interface, or you can now clone human protein and make those amino acid changes, and then you can apply this. So this shows that from this, what we have learned by studying proteins from different animals, that the protein, innate immune protein, peptidoglycan recognition protein in camel and porcine is far more potent than the same protein in humans. Therefore, the proteins from camel and porcine can be used for therapeutic applications to protect human beings. Does this make a sense? I asked them because they are the last. Does it make a sense? And the epidemiological data clearly say that camels don't have so common infections, that they have modified these proteins. And similarly, I think porcines also suffer less, unlike other animals. They're favorable. Cows and goats and sheep and buffaloes suffer most, because the protein, the same protein in them, is not protecting against these infections. So this is one simple indication. This has come from the animal kingdom and by isolating natural proteins. Okay, so this is one message. If you have appreciated, then we will quickly make another message very quickly. This is particularly relevant when you have this pandemic, coronavirus, when we were crying and we could not protect, but we could use this if we had this knowledge before. We were working on this system, but we had no idea when corona occurred, but subsequently some ideas emerged and then we found this very useful. This is protein, the lactoperoxidase, the heme peroxidase. So this is primarily to young boys and girls. When you look at a protein molecule, it looks a very complicated molecule to an untrained eye. This training is very important. To me, it looks very exciting, but to many of you, it may look like some mesh. You cannot make out of anything, isn't it? Except that Dr. Talwar, when he first saw some models of proteins, computer-based models, not actual structure, he said, this is a science I should pursue. So by looking at them, they're very exciting. Now I simplify this for you a little bit. Now in some kind of simple terms, I make it so this looks like some folded protein. These are helices, what you see cylinder, and you see the site here. Cleft, that is where these compounds bind. So cleft is very important. And if you see this more, the cleft is very important. So you must know that protein molecules are large globular molecules, but the binding sites are very specific cleft. And millions of proteins have different clefts, and that's how they distinguish which kind of partner they will have for interactions. More clearly, you can see even more clearly how the cleft is so deep and like this. And if I do it schematically, you can see the cleft. And from these amino acids, you can learn what is its chemical nature. So you learn everything. And once you know everything, you can do anything with this protein. If you want to inhibit its function, want to promote its function, you require this kind of knowledge. So this protein primarily is a large molecule, but the binding site, the action site is very little small here. I will not take much time, but should give you some idea. So, oh, I think I went in the wrong direction. Okay, so this protein, what it does, this basic thing you should know, that this protein requires hydrogen peroxide as a first step. See, all biological processes, you must know that, all biological processes in the living organism have pathways. Many enzymes have a single step, like trypsin requires a substrate and produces product, hydrolyzes proteins. This enzyme has two-step reaction. It requires hydrogen peroxide to create an intermediate, to create a complex with hydrogen peroxide, and then substrate thiocyanate binds, and then converted to hypothiocyanate, which is antiviral. So what you need for this enzyme, you need two substrates. One is H2O2 and one is this here, here, thiocyanate. This one and this one. With this, you produce this intermediate. With this one, using this intermediate, you produce product. So the schematic thing is like this. If this scheme was with you at the time of coronavirus, it required substrate SCN in good quantity. H2O2 is produced in the body in abundance, and then this enzyme will do the rest because this is expressed in high concentrations. If this enzyme is there, if this substrate is there, if this substrate is there, no virus bacteria can kill you. It has to be in the amount to be necessary to kill the virus and bacteria. So this is a basic thing. Now, this is something that was happening during coronavirus when we were not able to we were finding people were generating some data, making some observations. So first and foremost, you see that smokers were infected less. Can you believe this? You would have never believed in life that smoking is good. But it looks like good because you see that somebody made this observation that the smokers were not affected by this virus so much as the non-smokers. So you think about this. So don't make firm opinion based on social statements, but scientific basis you will see. And the next was that when potassium thiocyanate was given, thiocyanate the substrate, it didn't work. Smokers were happy, but thiocyanate administered in the form of potassium cyanide didn't produce the same effect. And then people said Japanese people are the best people in the world. They were not dying, they were not infected by coronavirus. You will wonder what is so different in Japanese people. Nice life, lifestyle. Lots of people said like you, they have a lifestyle, they laugh more, they interact more, they are friendly people. A lot of people talk like the way this boy is talking. So you see, but we see something else. The diet is good, but their diet contains a lot of iodide, the iodide-rich diet. And then potassium iodide also didn't work. And then you see children on and other people on nitric oxide support were infected less. These were the data generated in clinical lab by people. It's interesting, isn't it? If you had known, you would have taken nitric oxide in that time. And then, of course, patients on TB treatment were also not infected by this coronavirus. So the TB patients who were poor-looking people, they were happy during coronavirus. Others, non-TB people, were suffering more. So these are the things you should worry about, or you should be happy about. And then, of course, the last, not the least, the women were suffering less than the men. And that's another important thing. So these were the observations during Corona period, during the crisis period. These populations were very happily living, surviving, and we should see scientifically what is the basis. Otherwise, people will talk like him, the lifestyle, and then this and that, all kinds of things. So now we check this protein. What the question was that whether this protein was some way responsible this protein produces antiviral substances if it is given a substrate. So we will check this. So this protein has a binding site like this, filled with water molecules in normal case. Let's go to the next one. Then you check when the hydrogen peroxide binds to this structurally, experimentally determined. You can do a lot of postulation using computers and other hypotheses, but actual ways structurally determine this. So we check the structural binding of this. Hydrogen peroxide binds and forms the intermediate. Now, the next question is, when you said that smokers live happily, what does smoke contain? High content of thiocyanate. And they were smoking more thiocyanate, which is a substrate for this protein. And that's why when we check that thiocyanate binds to this very nicely here, and then the next experiment we showed that it converts this into hypothiocyanate, and that is antiviral, antibacterial. So that's why you know now, if you have to tell somebody, if there is a virus, to smoke more for a short while. Don't make a habit. So like this. Then I also said that potassium cyanide was given as a supplement, as a drug. It didn't work. It did not work because potassium ion of potassium cyanide bound at the site and blocked the binding of thiocyanate. The substrate was not able to be converted because the site is blocked by potassium. So one should use different compounds, not the potassium thiocyanate. Many times we just make salts of certain compounds without realizing, but that can have this kind of implication. Therefore, structure determinations are very critical to know what exactly is happening. Then the question comes, these Japanese people, why were they surviving most? And then you will be happy to see this data. This is recommended WHO. WHO recommends many things. Nobody takes it seriously, unfortunately. That's one problem. But in this case, they were very true that we should have followed. Indian daily consumption is 200 to 250. US and other Western countries, 150, very poor. And the Japanese, very Japanese. Down, where down? Here. So they were consuming more iodide than the Indian people, and Indian people were consuming more iodide than the European and Western people. And then you see the consequence of this. This is the consequence. This is I have written this, that this site was reporting things authentically claimed, and you see that how people were dying. There were other reasons why Indians were dying less, but Japanese were dying much less than these fellows. And Indians also had more iodide than the rest of them. So this is an indication. So iodide, if you took every day morning a lot of iodide salt, you will not be infected by these viruses and bacteria. So simple. You can practice next time. So then we show that what is iodide doing. Iodide binds to this, and you see this enzyme converts into hypoiodide plus IO. This is an evidence that this enzyme is converting iodide into hypoiodide. And that is the reason you are protected. That's why Japanese were protected more. And that's why Americans and Europeans were dying like anything. This was the first situation where Americans and Europeans died more than anybody else. Now here also you see potassium iodide has a problem. Potassium blocks this. So this, whatever was being observed, you are scientifically authenticating and proving so that in future you can recommend these kinds of things. Then next, this is something you should read about. There are colleagues in many places. There are some colleagues in IITs that they study mathematics, but they were giving solutions for this virus infection. They were bringing models. They were telling how the virus will go in the next month, how it was the previous month, how it will go next year. They were making all kinds of models. and models you can make, theoretical models, there's no restriction on this. So the slide primarily only jokes on this, that maybe this, maybe that, you can make anything. But actual thing is, this is what this lactobacillus system is the one which is doing the job, and nothing else is, you can assume, works. So the nitric oxide also same thing happened, that we showed that nitric oxide, it binds to this protein and is converted into nitric oxide, this here. NO2 is what is protecting you. So same thing you proved with nitric oxide. Then we tried many organic compounds. Some became as substrates, some did not. But one of the important ones was this isoniazid INH, the tuberculosis drug. It binds to this protein like a substrate. So anything which binds to this protein like a substrate at the proper site, it will be converted into product. You see, one thing you should know, you are students, enzyme reaction occurs. It converts a substrate into products, but the substrate must bind the required orientation in a manner, the binding site that's converted. If it binds there in some other orientation, it will not be converted. That's why many organic compounds may not become substrate, even if they bind here, but it has to bind in a required orientation. And then if it happens, it gets converted. So tuberculosis people were happy because they were taking INH. This is just to show that how in the structure this. And the last, not the least, is that estrogen. Estrogen is a hormone present only in humankind. And this hormone, when in some concentration in the body, this protein converts into an antimicrobial product. So if estrogen is present, the chances of your getting infection will be less. And that is why, that's because this is converted by this enzyme into a product which is antiviral. So this is a summary that this particular protein, if you supply substrates as a supplement or as a drug, then this will produce enough amount of antiviral, antibacterial product, and you will not have. I think you must be very happy you're working on tuberculosis. INS is doing many good jobs. So this is something. So these are some compounds we are listed here, which were being converted into this, and that's why. And this all has to be established. Everything we do with drug development, everything has to be scientifically tested and then trials made. But for many of them, trials are not required, so they're already in practice, they're already given. INH is given, NO2 is given, iodide is also in some form given. And so there's no problem. Estrogen is anywhere there. So all these things. So it's a very quick thing. And if we had known all this, we had established all this before the coronavirus, we might have not suffered so much. So the scheme now finally successful that you have your protein here. This H2O2 is abundance in your body. There's no shortage of this. You need to only make these substrates. SCN is not produced enough, so you supply these additional substrates, and then you produce enough of them, and then your viral thing disappears. And that's how we should make progress. And that's why animals I worship. Animals are like God for us. You see, many times people say that, why can't you clone and express proteins and do away with this and do structures? But there are many things, many of these ligands we observe when we isolate natural proteins. We observe SCN in the OSC and SCN in the protein because we isolate it from the natural source. So studying proteins by isolating from natural source can give you many new insights. And also, when you saw the previous camel case, those mutations, you cannot imagine mutations if you are working on a cow protein, same protein, and you clone and express, or you just work on cow, you cannot know it. But if you do it from different animals, they would have behaved differently in different times because of these infections, and they changed their proteins by introducing some minimal mutations and made those proteins far more potent to fight against infections. So this is essential, Taru, to support your NIAB high quality science in a simple way, that how this can be done. So this is just to thank you. My focus was these lot, so that the future, India's future is young generation. People say that all those people who train young people, that is the best industry, that's the best raw material India has. And this raw material is wealth, and this raw material will determine our future, our economy. So I think that's why my focus was on you. Many senior scientists, very knowledgeable scientists and experts, they will find this very preliminary or very elementary. But I think if this lot says wonderful, I'm so delighted. Thank you so much. I think that the story goes like this. So maybe I will be happy to respond to any kind of questions and comments. And if anybody can ask, yeah.
Wonderful to see you how nicely you converted your lecture into a school class, and I'm pretty sure all the school kids understood, and today they got inspired to do possibly science in future. So I'll call my colleague Dr. Madhuri take over and facilitate the question.
She warned me that if this lot is not happy, your lecture is useless. So I directed this, and I hope they will not say negative. Questions and comments, because the way you learn is one of the reasons why one should give talks. We learn a lot by giving talks, by learning from questions and comments. So please make questions and comments, and all questions and comments are there's somebody there. Yes, sir. Yes, madam.
Don't have that.
Okay. So she is asking a question, why certain proteins change and improve their quality and then develop a potential for therapeutic application, why others not? I think it's a challenge given to things. There was a guy, I tell you, one German colleague was visiting me some years before, many years before, when we were not using this Bisleri, this water. He came with 3-4 bottles in his pockets, and he came to my office. I said, what is this? I drink this water. I have to survive on this. I said, okay, survive. Then he wanted to go and visit Jama Masjid area, the area of Delhi, which is, I think, highly polluted. He went there, and he saw people jumping and playing without anything and drinking anything. He felt very stupid of himself. He said, they are also human beings, I'm also a human being. If they are surviving, why I am not surviving? Why I need this? He threw bottles and came back. He said, I don't require bottles now. So why? I want to be like them. So it's a question that when you are exposed more to these organisms, and the proteins that are in the body protecting against them, they also threaten the way these organisms modify themselves. All organisms, we are also an organism. So those proteins change. In case of camel, I used to think that we have that, you will correct me, of course. I used to think that camel lives in a hot environment and no bacteria and virus can survive there. It was not right. The powerful bacteria survive. They also become stronger in desert environment. So they are stronger and they are attacking animal. So animal has to develop a stronger response. And that's how those proteins are modified. So our many concepts we change when we actually see things. So initially, you have some notions. He said that smoker, he was laughing at smokers. How can smoking be good? It's banned, it's not good. But to fight virus bacteria is very good. Yes, sir. Thank you. Is it okay? Yes, sir.
I have two questions basically. So first part is related to the structure you showed that very similar and they make the
Oligomeric state changed.
So it's something new to see that when
Yeah. 2 amino acids changed. Yeah, there is one very general way to say that whenever proteins on the surface, you see the surface residues are mostly these polar residues, arginine, lysine, glutamic acid, glutamine, the random size of their side chains. If you can introduce some prolines, proline is only one and it packs into a dimeric state much more. So 70% of dimers are formed. Prolines are then phase. The propensity of dimerization improves if you introduce proline. In the human case, this is arginine and histidine. In the camel case, both are replaced by proline. It became dimer.
Anybody has shown in experimental condition that it's possible that it can be inhibit virus entry or inhibit by using molecules.
This is well known that these products like OSC and hypothiocyanate, hypoiodide, people have done an experiment. They kill these organisms.
The coronavirus, it was not in culture.
Yeah, no, it's not done, but I think it can be done. But that is the beauty of the innate immune system. It is not dependent on a specific virus or bacteria. In general, this is how it is innate, this is how it's the first line of defense. It kills every organism because nature is that way. But you are giving a bad environment to these organisms to die. So whatever virus, whatever bacteria, its effect will be because it's a kind of milieu you are changing. It's from their proteins and they find and they do this general mechanism. Where this? Okay. I'm only afraid of microbiologists. And if you are not a microbiologist, I'm very happy. Okay. Of camel and human. It's identical. It's not something like 0.5 angstrom. 0.5 angstrom. They're identical structures.
When we are seeing the structures which are produced by the enzyme,
This is
there we are having a family of structures, and they are taking care of the dynamic, the motion of the protein. On the other hand, X-ray, only single snapshot. How you will take care of the motion? Because see, these are the, what to say?
okay. I understood your question. You understood the question? This question was asked more than 60 years before. What happens to a structure? The structures by crystal reactor when the protein is in the crystalline state, so-called solid state. It's not exactly solid. It's not sodium. You've seen sodium chloride crystals? You have never eaten sodium chloride. Sugar crystals? Sugar crystals, you see, if you put this, nothing happens. But if you touch a protein crystal, it disappears. The protein crystals have solvent content up to even 90%. 30% to 90% solvent content is found in protein crystals. So protein molecules in the crystalline state are not actually a solid state. It is like putting molecules in the solution state with one orientation, with relatively fixed orientation. And there's so many arguments on this. I think we should make argument at some time. You see, protein molecules, when they fold, the basic principle of protein folding is maximize the attractive interactions and minimize the repulsive interactions. This is basic. And these interactions come from large number of interactions provided by amino acids, main chain and side chain. So by and large, protein structures are very stable. There might be minor variations. And that's what NMR people do. And that's what people in computational biology, what they talk about molecular dynamics do. But these are very fine things. That grossly, the structure is not different. Structure remains the same. And grossly what we are seeing, how the interactions between two molecules occur. These variations are not going to hamper these interactions, but they are based basically on how complementary the binding site is and what amino acids provide the chemical interaction support. So basically what you have is structures from max range is the right model to pursue any design or understand any mode of action involving these proteins. So it is like this. So that is fine. Some, because they are in solution, they will be changing some orientation and some parts on the surface. In some cases of proteins are flexible, and they are flexible, not just like that, they're flexible because they are important in some certain protein-protein interactions. And this flexibility is required, but when they bind, they get steady. So there is no issue or lack of knowledge by this kind of event in terms of understanding the mechanism of action, or in terms of designing compounds to block or promote their functions. So it is like this. So NMR, XA, complementary. Both are friends. Once I complained to one of the NMR guys, your famous friend, are you soon? Yeah, so the problem is that X-ray thing is continuously growing and NMR is reducing. So it's real basic science which is necessary for us to understand protein structures, their functions, and to design drugs. There is no question. Even the cryocrystallography is emerging, but this will remain. This will never go. So this is in favor of this subject. Hello. You're asking on behalf of them. Other, right.
So that we lack the interaction site or we lack the subunit system.
That's in human protein, you change two amino acids. One is histidine, one is adenine to proline. It becomes a dimer. And this demand came because camels and porcine had the bigger this pathogen load. So over the period they changed. See, all of us, all living organisms are natural labs. You do in the lab these mutations. The nature also does it to perform better, to improve. And when she was asking, wherever there is a threat, the participation to improve, these changes occur. So these specific changes occur. And that's very interesting.
We do have those other subunits, but we lack the interaction.
No, this protein in human is monomer. Others is a homodimers, homotrimers. No. It's the same protein in the form of dimer and trimer. That. Antibodies. There's one question people would generally ask. that if you change a protein, it may become antigen. It may cause antibody production, but it's the same protein. And the reason was that when you have these changes, they become part of the interface of a dimer. So they are not really invoking this antigenicity at all in this case. But this is one common argument comes to mind. I agree. Okay. I'm not the monitor, she is the monitor.
That is problem that we have in the and which are mentioned in the addition to the bacteria. If we apply these are compatible.
He was asking the same question. It is compatible.
And first of all, you know, telling that we apply the substrate for the isotyrosine.
Yeah, the last one, last protein.
What the enzyme is also important.
Production of enzyme increases when there is an infection. It goes up very much up. So enzyme production in response to infection is more, but there is no initiative for thiocyanate to be more. There's a metabolic products that is controlled because of infections. So that has to be supplied. And H2O2 anyways produce much more. So this substrate, thiocyanate, has to be given from outside. It's difficult to give from outside. Think of some design substrate, and that can become a medicine. Like INH also is a good option, but you can make better options. INH incidentally.
Maybe the same for the same substrate. So it may be
When you are in a crisis, when the protein is expressed so much, just think of one thing that provides substrate so that can protect you from dying. Think afterwards what other effects it will have. It may have, it will not have. Yes.
So in your one slide, you mentioned that.
That side?
Yeah.
Okay.
So that iodide consumption will it will be.
Sure.
We all know that iodine is very important to, you know, regulate the thyroid.
Right.
So is it that iodine,
Yeah, this also binds. So far, we knew that iodide is required for thyroid problems. There is also an enzyme of the same family, thyroid peroxidase. Thyroid peroxidase, lactoperoxidase, myeloperoxidase, eosinophil peroxidase, they are all the same family. They are members together. So additional iodide can be used there as well. There is no problem. That's one thing. But at a given time, when the concentration of the particular enzyme is much more, it will consume much more thyroid. It is available in the environment. And I didn't say that. When we look at the structure of this enzyme, several attachment sites with thyroid can attach with low affinity. So it carries a lot of thyroid ions anyway, all the time. And when there is an infection, it uses them as substrate. Same nearly identical structures, thyroid peroxidase. So it will do that job also. That is one question. I think that perhaps one can address in some way later. But currently, that works for this purpose. Maybe that you can determine by dosage, quantity, all those things, and see that. So it doesn't cause that kind of problem. That way. That means. Right.
well, we might what protein whose structure like template is available. So if we work with that protein in this kind of doing this kind of possible, what the protein
Okay, so his question is understood by all. He's saying there are some, so you must be hearing word this artificial intelligence. This is artificial, it's not real. That's number one. This artificial intelligence is dependent on the quality of databases you have. And the AlphaFold he talked about is also based on the database we have on protein structures. Now, if the database, which is because of the experimental data, is not good enough, AI will be meaningless. So AlphaFold is interesting, it's a very good development, but it cannot give you everything. There are many situations where proteins it will not give you idea about binding and interactions, natural proteins. Some ligands may be already there. So actual structures have to be determined by experimental method only. But this is real that number of labs doing structures is reducing. It's not a good idea. It is because small, as you said, every protein cannot be crystallized. People try to do crystals, prepare crystals. The rate limiting takes time. We will move to other sites which are faster. But this will remain. This is the real science. It will remain, whatever number of people may be pursuing.
the question is potential because this is the amino acid driven. So can we is there any tool or something?
With your knowledge, when your knowledge is growing, you can do many things like this. But you require knowledgeable people. If you are doing purely based on silicon things, you may make errors. But if you understand these structures, you examine these structures with your eyes and you understand, you can predict these things much more accurately. So that's where I always insist that there are lots of things when computational biologists, I hope there are not many computational biologists here who will be angry, who will become angry. Computational biologists, they are very much dependent on what computers do, some numbers, and many numbers don't make a sense to common people. But what is required together with that? Full understanding of the protein structures. And that will check, that will tell you what is right, what's wrong. So with knowledge and other things, you can predict. You decided you are the last, very good. That is good idea.
We generally make a lot of dynamic radical microbes.
Very nice.
So the whole mechanism comes from the dynamic and very good.
Very right.
So another thing is most of the previous products not very and they told that technique is available. So when does the innate immune system know that how is the dosage decided in the organism? It is channel and the microorganism infecting or the pathogenic channel. Both of the worried for a phenomenon.
Okay. 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 He is asking a question that is the innate immune system so bad that if it starts, never stops? Oh, how does it stop? When there is a demand, it will keep giving. Otherwise, what happens that all these products it is making, they bind back to this and block its binding site, so it stops that way. So when you are not requiring any more these products, they go back and bind to the binding site. And in the structure also, I showed that all these products, they bind nicely to the substrate binding site when they are not really, when there is so much negative environment outside, they will go and sit inside the binding site. And that's how it blocks the enzyme also, and it resides there. So this happens all the time.
So what went wrong with COVID? You can see two sides of COVID.
Mm.
There was actually the immune system that even was the immune system that was poor.
What went wrong with COVID? He's asking. Has anybody any answer? I think that what we talked today, this was not known before, number one. So this could have been some help. But what went wrong is, I think this virus has a very high affinity receptors in your body, and it enters very rapidly. And obviously your antibodies take time to emerge after some days. And within that period, it makes the situation very worse. And all our innate immune fellows, they had some role to play, but this was perhaps too much for them. But we can strengthen innate immune system next time to fight it back if we had known all these things. If we had consumed more iodized salt, if we had smoked, then also. And if we had certain things, that's the idea. Thank you so much. Last was not this. Okay. They bind to the cell wall, these compounds I showed, LPS, LT, and then to the next component, which hydrolyzes that stuff. No, it is not doing that. The one is that some molecules, there are 4 members. One molecule just binds and sequesters. And what it does afterwards, I don't know. Perhaps you can tell. But there are other 3 components. They bind and they hydrolyze also. Direct. It's a direct action. These are all direct action proteins. No. There's no signaling in that involved. So, madam, we are ready. There is one more. Huh? Something. We forget things so often. Okay. Okay. Yeah. No. It's really complicated. I sometimes wonder that how does the system work? I would say it should fail all the time because there's so many complicated events are going on. So many millions of molecules intermingling. It's amazing that it works. But it works because of the specificity of these proteins. They perform specific functions. But there are many instances where these things go wrong also, wrong mutations also occur. And there are many reasons why they occur. You know, we take millions of compounds inside our body, lots of things get affected. But in this case, you have clear observation that what has changed and has changed for good, so we can exploit this for applications. It is like this. Lots of wrong things can also happen. But this is a very nice observation that the dimer trimer became better binders to the cell wall molecules than monomer. I feel only one thing. As a scientist, we should do small experiments and continue to build upon them and not to worry about complications. A day will come when we will understand everything about these proteins and we can answer these complicated questions. So that side that many complications are there, many things can happen, but we should not think. So scientific research, pick up anything very small, sorry to say differently, and just build it upon that, and you will grow accordingly. And I think complications we can tackle later. I feel like that. Oh, this so.
Thank you for the question.
That is this people want there.
No, this is for you.
So I say this, like this that we have. Yeah. Okay.
Yeah, so we decided that our books are the best friends, and I think
Is the conclusion. Okay. How many books are there? But there are
we
more
request
still.
the teacher. We go to the library.
Okay, I will allow you. Thank you so much. You are feeling nice? Are you happy? You are the other teacher.
Thank you.
Thank you so much.
Actually, do it here.
I can sign it. You can sign? This one. Sign it here. And here. Okay.
Thank you.
Anything else? Thank you so much.
Very good.
Thank you very much.
Okay, so all are invited to join for cup of tea and some